you have actually what is called resistance. And these are all of the means developed by the host to actually directly attack the pathogen and try to get rid of it. And of course, in terms of innate immunity, you can think about antimicrobial peptides that you find in all kinds of living organisms. And then you have also complement, cellular immune response. I mean, since Mechnikov, we know that phagocytosis is very important. And then in the case of the adaptive immune response in higher vertebrates, you have antibodies and T cells, and also you can think about reactive oxygen species. So this is basically the business of immunology and immunity. But there is a second aspect that is much less understood, and that I call myself resilience, and that you can find in the literature under the vocable as disease tolerance. And this basically is this capacity that the organism has to be able to endure and to some extent repair damages that are inflicted by microbial virulence factors, or like you can think about toxins, performing toxins, outer membrane vesicles from bacteria, and actually, or it can be also inflicted by the host immune defenses from the host. And this is actually very important. It makes actually an interface between homeostatic physiological responses and infection. Okay, and in the talk, I will illustrate some of these key points between resistance and resilience. So, in terms of immune response, generally, we have some general principles. In many cases, we need to have an inducible immune response because it's energetically costly, and you want to trigger it mostly only when pathogens are encountered. And again, the distinction between self and non-self is, of course, critical for this. What we have is then after once you have triggered this immune response, you're going through intracellular signaling pathways, for instance, NF-kappa-B that you find in flies or in mammals, and also MAP kinases pathways such as JNK pathway, and actually C. elegans, nematode worms, and Rhabditis elegans, is lacking NF-kappa-B, but is relying instead a lot on the P38 pathway. And then, of course, we have all of the level of effectors. Antibodies, for instance, in the adaptive immune response, as discussed in the previous slide, and in the case of of innate immune responses, antimicrobial peptides. So I just want to highlight the basics of an immune response whenever you're making actually an injury or you're injecting microbes into the fly. What you get are immediate early responses, which are basically based on proteolytic cascades that lead to coagulation, and also to an arthropod-specific response known as melanization, in which you activate phenol oxidases and you produce melanin as well as other antimicrobial molecules that will act on the invading pathogen. So you also have a cellular immune response mediated by hemocytes called as plasmatocytes, and then we have also a very important systemic immune response, whereby once you have detected the infection, you activate one or two type of NF-kappa-B pathways known respectively as TOL or immune deficiency pathway in these fat body cells, and these fat body cells are a mix in insects of liver cells and also at the same time of adipocytes. And you can release actually, depending on the triggering activation of these pathways, of lots of different antimicrobial peptides. Most of them are regulated in response to gram-negative bacteria through the immune deficiency pathway, and you have diptericin, cecropins, or atacins. And in the case of some gram-positive bacteria and fungal infections, then you have production of drosomycin and mechnicovin. And this can be actually quite important in terms of quantities, because in the case of drosomycin, after 24 hours, you're going to concentrations in the hemolymph of 0.1 millimolar. So it's really a very strong, 100 micromolars. So it's a very strong investment for metabolism also. Okay, so then we have this collection of antimicrobial peptides. we know that the toll pathway is actually very important for defenses against fungi and some gram-positive bacteria. And we have some kind of discrepancies. So one of them is that defensin are the only antimicrobial peptides that are known to be active against gram-positive bacteria. And the problem is that when you're getting rid of defensin, either by mutating the IMD pathway or making direct deletion, you do not see much of a phenotype in terms of host defense against gram-positive bacteria. Also, we have no antimicrobial peptides that have been described that are able to act on pathogenic yeasts, like Candida albicans or Candida glabrata. And finally, we know that there are many genes that are regulated by the toll pathway, and this is at least 150 genes and probably more, just a matter of thresholds. So one of the questions we asked when we really started this new lab in Guangzhou was, can we do something more to understand fungal infections using our favorite model organism, Drosophila? And actually, one part of the answer is coming actually from biochemistry. And just to remind to you, actually, when Philippe Bullet and colleagues tried to isolate drosomycin, they started from extract of 50,000 flies that had been challenged one by one. Okay, so it was a huge work in a differential screen. And then Philippe Bullet started to experiment with mass spectrometry back in at the end of the last millennium. And what he found is that there were inducible peptides that you could find. And actually later work showed that there are two different categories, major categories that account for these peaks. One of them actually belonged to a family of peptides called bomanin, and these have been really, so the three first peptides all share the same domain, the so-called bomanin domain, and then Steve Wasserman actually extended the whole concept of bomanins and found out that there are 12 genes belonging to this family. 6 of them actually belong to the short bomanin family. And actually, what you produce is just a peptide of 16 amino acids that correspond just to the bomanin domain. And it's so highly conserved that between these 6 short bomanins, there are at most 1 amino acid differences in terms of primary sequence. Then you have a second category of bomanins. There are the so-called tailed bomanins, in which you have an extension at the C-terminus that is extending from 20 to 80 amino acids. And finally, there is a third category called the BCP toll, because they have 2 bomanin domains with a linker in between, and there are 3 such bomanins. And what was a very interesting finding is that actually out of the 12 family members, 10 of them are actually in the same, on the same locus at 55C in 10 kb. Okay, so they are all really concentrated. So the second family of peptide is actually called baramycin A, and this has been worked on in our lab and also in the lab of Bruno Lemaitre by Mark Hansen. And this was for a long time a mystery. So we all know that these peptides are derived from a common precursor that used to be called IMPPP. And you can see that all of these red proteins are short peptides or longer ones, 22 and 24 amino acid domain. Okay, so then what Steve Wasserman did once he had identified this locus, he actually generated a mutation that was deleting this whole locus. So you're just destroying the 10 bomanin genes, and this mutant that we call 55C, bomanin 55C deletion, is actually this light blue curve. And you can see that as expected, these flies are perfectly resistant to a challenge with gram-negative bacteria, such as Enterobacter cloacae, whereas immune deficiency pathway mutants are very sensitive to this challenge. And what was very striking is that when you switch to Enterococcus faecalis, a gram-positive bacterium, what you could see actually is that the 55C deletion were as susceptible as toll pathway mutants to this challenge. Okay, which means that you had actually most of the functions, effector functions of the toll pathway are going through these 10 peptides. And the story was also very similar when we looked at Candida glabrata. And again, the 55C deficiency phenocopies to a large extent the toll pathway mutants. And finally, very similar observations were made using a filamentous fungus, Fusarium oxysporum, and you can see that they are dying at the same rate as Tolpafo mutants. So which means that the 55C bomanins are really mediating an important aspect of the host defense against these different pathogens, gram-positive bacteria and fungi, so yeast or filamentous fungi. So we have actually been dissecting genetically this locus, and I will just show you very few of the data. And for some of them, we managed actually to get CRISPR-Cas9 mutants. It's easier for the longer ones. The short ones are so conserved that it's very difficult to get good mutants in them, and we have failed for many of those. And we have also a second strategy, which consists in overexpression, and I will illustrate this a little bit later. But what was very striking in the case of bomanin T1, whenever we are deleting this peptide gene, we find that the mutants are as susceptible as Tolpafo mutants to a challenge with Enterococcus faecalis. And as shown previously by Professor Jiao, we found actually that in the mutants, there is actually an increased microbial titer, really suggesting that bomanin T1 functions in resistance. Okay, so it's really potentially acting as an antimicrobial peptide, although we have no in vitro evidence to say this, and we still need to work out the mechanism by which it's active on Enterococcus faecalis. So what was interesting is that we also found that BOM-T1 mutants were susceptible to an infection with an entomopathogenic fungus called Metarhizium robertsi. And in this case, when we're doing this infection, the fungus is killing all the time, but the Tolpafo mutants are dying somewhat faster. And what we find in this case is that actually the BOM-T1 mutants are dying as fast also as the Tolpafo mutant. But in contrast to the Tol mutants, you can see that in total mutants, you have an enhanced and increased microbial titer, and that it's starting really towards the end of the infection between day 2 and day 3. And then after at day 4, actually, you no longer have any Tol mutant flies living. And then we did not detect this phenotype with BOM-T1 mutants, suggesting that possibly BOM-T1 might be not functioning in resistance, but more in resilience to this type of infections. So to sum up at this stage, we have these two facets of BOM-T1 functions. One of them is resistance. So it could be acting as a potential or contribute as a potential antimicrobial peptide against Enterococcus faecalis. Again, we don't have the proof for this. But what we found also is that BOM-T1 is actually able to prevent the dissemination of Candida albicans in the body of the fly. So it's also able to work against an eukaryotic pathogen, namely this pathogenic yeast. And at the same time, we have also this aspect of BOM-T1 function in resilience, where actually we find that it's able, as suggested for Metarhizium robertsi, since we don't have an effect on the microbial titer, it could be really acting in resilience and possibly in protecting from mycotoxins. And actually, we have found a very good phenotype with this mycotoxin from Aspergillus fumigatus called restrictocin, and I will show you more of this in the next slides. So before this, I just want to switch a little bit to one of the strategic decisions we took when we started the work in China. It was really to focus on fungal infections because, of course, fungi are really widespread pathogens and they are attacking plants, invertebrates, and vertebrates. In humans, they are very important. Collectively, fungal infections kill about 4 million people per year, a little bit less. And actually, you find that 6.5 million patients worldwide are suffering from invasive or chronic fungal infections. So it's actually something that is very prevalent. And of course, it's economically costly because you need to treat all of these patients. And then, of course, most of these fungi are actually acting on suppressed or immunodeficient people, like people suffering from AIDS, from cancer. And what has been very strong illustration is during COVID, where many people were not killed actually by the virus, by the concomitant fungal infections, by the black fungi, for instance, in India, also a lot with Aspergillus fumigatus. Okay, and then you really have dependence on the origin. So some of these fungal pathogens are prevalent in poor countries. So that's the case of Cryptococcus. And whereas others are more reflecting advanced medical state where you're using a lot of immune suppressor drugs, and you find invasive candidiasis and aspergillosis being more often found in rich countries. Okay, and of course we have also you have local fungi such as Stolomyces manifae in the Southeast Asia and in China, and we've been working also a little bit on this. And what I really want to make the point is that there is a link also with global warming, because what you get is higher temperatures, but also quite often you get typhoons, you get widespread floodings. So right now in Thailand, it's a disaster. In the Philippines, it's also a disaster. And this was also the case in Guangzhou. I think that there was some strong floodings in Guangdong this year. And every time you have floodings, you get fungal colonizing the walls of houses, and basically you will never get rid of them, and you will be exposed to all of these volatile toxins. So it's really a major health problem that is developing. And what is really compounding the problem is also that you have only 3 drug classes that are effective against this eukaryotic pathogen, and they are expensive, toxic, and also widely used in agriculture, such as azoles, which means you're selecting for resistant strains to which human patients are then subjected. Okay, so, and then of course the field is understudied, and I would really say that we need more money to work on those, as usual. Okay, so what are the specific features of this fungi? Actually, what we find is that in the case of plant pathogens or entomopathogenic fungi, they are secreting hundreds of proteases. Okay, that's really prevalent. So something like Metarhizium robertsi has 800 proteases that are being expressed during invasion phases. And they are also master of secondary metabolism through different synthetic modules. And that's also one important aspect. And I want to illustrate this with this masterpiece from Matthias Grunwald that is actually exposed right next to Strasbourg in Colmar. And this triptych as actually was part of, was exposed in a hospital from people suffering from sun and furnace fire. And the idea of the painting was to show that even though they were suffering really terrible pains, it was much less than what Christ had been suffering on his cross. So that was the point of the painting. And actually, it turns out that many of these people were actually suffering from infection contaminated rye by this fungus called Claviceps purpurea. And some of these virulence factors are ergot alkaloids, and they can have very strong effects by promoting a strong vasoconstrictions and actually the limbs of patients who are rotting. So these were for the strong exposures, but there were also some neural manifestations. And for instance, it's been very well described that in 1518, there was a dancing epidemic in Strasbourg, so my hometown, where people would actually be dancing for days until they would collapse in exhaustion. So it was very surprising, and was linked really to poor when the years were very wet, and that there was a lot of casualties. So to go with Drosophila, we decided to study on to focus on Aspergillus fumigatus. And what we found actually is that Aspergillus fumigatus mutants are not able to kill wild-type flies, but as few as 5 conidia will kill tall pathway mutants, like in this case MYD88. And what was very striking and unusual, that usually when you activate the toll pathway, again, it's required for defense against fungal infections. Drosomycin is usually strongly induced, and this was not really the case when we were infecting with Aspergillus fumigatus, as compared, for instance, to a gram-positive bacterium. And that was very surprising and confirmed by mass spectrometry. And also, in contrast to what I showed to you with Metarhizium robertsi, when the flies were dying, there was no increase really in the fungal burden in the diseased flies. And what turned out is that actually melanization is important to prevent the dissemination of the fungus. So you can see here in the Toll pathway mutant fly, the fungus remains confined to the site of injection. Whoa. Okay, thank you. And in the case of MYD88 flies, you can see that the fungus is disseminating. And actually, when you're blocking melanization, you have a much higher titer. So what this really states is that what is the function of the Toll pathway then if it's not required to directly fight Aspergillus fumigatus? And what we found, to make a long story short, is that actually the Toll pathway is required for protections against several toxins secreted by Aspergillus fumigatus. And in this talk, I will focus on variculogen, a secondary metabolite, and also on restrictocin, a ribotoxin. So basically, the idea was just actually to buy these toxins from Sigma. And so when we injected them, we could see that wild-type flies were not dying at all when we were injecting restrictocin in different concentrations, but Toll pathway mutants were really dying very fast from this. And actually, restrictocin were probably pushing the system because it only contributes very mildly to the total virulence of the fungus, because when we have fungi that are unable to make restrictocin, you see that they are still pretty virulent in Toll pathway mutant flies. And we can actually check using biochemistry. So this is 28S RNA using bioanalyzer. So these are wild-type flies that have been injected with restrictocin, and you can see that your 28S RNA is perfectly fine. But whenever you're using Toll pathway mutants, you can see really that when you're injecting restrictocin, you're actually destroying and cleaving this 28S RNA and actually blocking translation from the host. Okay, so in the case of variculogen, we have a similar story. Variculogen is somewhat toxic, but it's much more toxic to Toll pathway mutant flies as compared to wild-type flies. And again, variculogen only contributes very mildly to the overall virulence of the fungus in flies. And if we can start the movie, what is very striking is that when you're injecting the toxin, what we see actually, can you start the movie here, please? Yeah, okay. The flies are actually immediately paralyzed. And what you can see is they stay on the back and they just have some movements from their legs. And this affects the wild-type flies as well as the Toll pathway mutant flies. But what is important is that after a period in this experiment between 15 and 30 hours, you can see the wild-type flies recover from this paralysis, but which is not the case from the Toll pathway mutant flies. And what was interesting is that if you overexpress the Toll pathway using a genetic trick, then you get partial protection from these tremors. Okay, so then we actually tried to understand what was going on. So again, 55C mutants are also susceptible to Aspergillus fumigatus. And then the idea was, since we didn't have all of the mutants, was actually to try to make a complementation assay in which we were expressing each single bomanin into the context of this 55C deficiency. And again, yeah. Okay. What you can see, wild-type flies are the most susceptible to variculogen. And what is very important in red this time, the 55C mutant flies are sensitive to variculogen as they are to restrictocin. And here we have some controls. But when we are overexpressing, in the case of variculogen, BOM-S6, or in the case of restrictocin, either BOM-BC1, BOM-S3, or BOM-S6, we can get a partial rescue from this sensitivity to restrictocin that we do not see when we are actually overexpressing the other bomanins of the domain. So, what you could find out also is that we overexpress, activate the Toll pathway in the nervous system. We get a protection from these tremors. And also in terms of survival, sorry, and trying to get to the next slide. Okay, what you get is that actually whereas the flies are dying, even the wild-type flies from variculogen, when you're activating the toll pathway in neurons, you get a very strong protection, and none of the flies are dying. In the case of the overexpression of BOMS6 in the nervous system, we do not get any effect on tremors, but we do get actually an enhanced recovery. So the flies recover much faster from the tremors. Okay, and then in terms of survival, as for the toll pathway, when we overexpress BOMS6 in the nervous system, we get a very good protection from this challenge with restrictocin as compared to 55C mutant flies. So BOMS6 is actually, as I was showing to you, able to protect to some degree from the action of variculogen. And BOMS6 is known to actually target a maxi potassium channel known as Slowpoke in Drosophila. And we actually have the structures that has been published, and with variculogen acting directly on this receptor. So BOMS6 must be in some way able to displace this binding at some point. And on the other hand, restrictocin is also being protected by BOMS6 overexpression. And in this case, we're talking about a protein that is acting in a totally different manner by actually cleaving 28S RNA. So it's really something very surprising that one peptide can do the two things at the same time. Okay. So then we were trying to find out what was the role of the toll pathway in the nervous system. And actually, one paper, very interesting, showed that the toll pathway was very important in the homeostasis of sleep. So basically what it's doing is that following stimulation and production of its ligand by dendritic cells, so actually glial cells, you activate the toll pathway, and this is required for sleep homeostasis in this R5 neuronal network. And what is very neat is that many people are studying neurobiology of the fly, and you can see here very neatly these R5 neurons being expressed here in this ellipsoid body in the brain, where sleep homeostasis is thought to be regulated. But we see that these R5 neurons are also expressed in the ventral nerve cord that you can see here. And what Bo did was actually to silence by RNA interference toll or MYD88 only in these R5 neurons. And then what he found out is that these flies were getting as susceptible to variculogen, that I'm not showing it on this slide, but also very susceptible to restrictocin when you silence toll only in this neuronal network. And we made a converse experiment by actually rescuing toll activity only in R5 neurons. And then we got a partial sensitivity to Aspergillus fumigatus. Okay. So one of the things that we can get is Bo did this very crazy experiment. So we took headless flies, and when he's injecting PBS, you see that they are not moving. But whenever he's injecting actually variculogen in these headless flies, you can see that they are still very reactive and showing some tremors. And from this, we conclude actually that the toll pathway, if I can go to the next slide, is actually probably more required, not for this immune function of protection against toxins, more in the ventral nerve cord. Okay, yeah. Okay, so to sum up, we have what I've shown you is that resistance against Aspergillus fumigatus is mediated by myelinization, that the toll pathway protects against the action of secreted mycotoxins through specific abomanins, and that we have these two functions of the neurotoxin of toll against variculogen. One unidentified effector is able to protect against the action of variculogen, whereas BOMS6 is able actually to enhance recovery from variculogen action, and probably using this R5 neural network. Okay, so I will go very rapidly on this because I'm going over time. So we have also worked on baramycin A and other effectors, and this work has been published already in the case of baramycin A. And what we have found is that baramycin A is protecting actually against the neurotoxins from Metarhizium robertsi, and that at the same time, baramycin A was able to protect the fly from the action of an enterocin, so a bacteriocin produced by Enterococcus faecalis. So these are effectors from one eukaryotic pathogen or from one prokaryotic pathogen, and the same protein, again, is able to do these multiple functions. And we have this also in the case story is a little bit more complex, and I don't have the time to go over it, but we have also two further peptides that are very important in those defenses against this destructin and possibly enterocin. And we found actually that this baramycin and these two peptides are important for protections against the protease from the Serratia marcescens, and actually likely also from Pseudomonas aeruginosa, and I will not have the time to go too much into it. But what is really striking is that we have, again, toll pathway effectors able to provide protection against really a variety of secreted virulence factors that have distinct origins and biochemical activities. In some cases, we have some hypotheses, but I think we're still far from understanding what is really going on, and we're still working on this. And the last point I wanted to make, if I can manage to get to the next slide, to the next thing, is that there are human patients, especially all of the people who have been suffering from tuberculosis. Then they develop aspergillomas because they have these vast cavities in their lungs. And these people, it's more than 2 million people worldwide, they are really exposed to Aspergillus fumigatus chronically and to the secreted virulence factors. And the question is, I'm not sure that we have any adaptive immune defenses against mycotoxins, because many of them are small metabolites, and I'm not sure that we can derive antibodies. We can certainly derive antibodies against restrictocin. It's a cause of asthma in young children. And what is also an important question that should be addressed at some point is, do we have innate defenses against actually these mycotoxins in France? No, sorry.
It has now become apparent that in some of the fundamental principles we learned from the science apply to higher versions of God, including human beings.
Okay, so this was just the conclusion given by Professor Nusslein Vollard during her speech during the Nobel Prize banquet back in 1995, in which she was really highlighting that all of the work done on Drosophila melanogaster is quite often relevant to an understanding of human biology in the long term. Okay, and with this, I would really like to conclude and to thank all of the people who have participated in the work. Sorry, I have no control over this. Can we go to the next slide, please? Okay, and I really want to thank all of the people who have been working, especially the people working on Aspergillus fumigatus, work initiated by Ray Chu, and the work on the effectors, especially Xu Ping and Jian Chong, and all of these people. And I really want to thank them for their hard work and for their enthusiasm, and of course all of the founders, and this wonderful opportunity we had to work here in China. I thank you very much for your attention.
Dominique. Okay. Regarding
restrictocin.
You said that R5 neurons and the ventricle are essential for
the protection against the toxin.
Yeah. So why? Well, I just did not get it. So one is the central nervous system, another one is it's also central nervous system, it's not in the brain, the ventral nerve cord. So are these two parts kind of connected to each other or communicate with each other?
Yeah, this we don't know. I mean, we've been trying to play a little bit to see whether we have an impact on restrictocin on sleep homeostasis, but I don't think we went very far with this. And it was difficult to test, even though we have the equipment in SFHI, but students didn't really have the time to go into this.
So the toxin, it acts directly on the neurons or through other molecules?
Well, I mean, story is actually complex because we have been also doing large-scale screens, and we have found molecules that are required in neurons, and others are required in glial cells, depending on the toxin you're using. So I think we're probably just scratching the tip of the iceberg in terms of understanding what is going on. And for the time being, we have more focused on these effectors. And one of the critical questions I'm hoping to find the answer when I'm going back to Guangzhou tomorrow is whether you actually have something, like the R5 neurons, are they just one neural network? Or are they acting through a systemic response to the rest of the body? And I think it's one of the critical questions I really want to have the answer to. Or are these R5s, the peripheral neurons, the ones that are really regulating movement? It's kind of an interesting question, and we don't have this answer yet.
Yes. Yes, Dominique, I'm always curious because since for example, in one fungi, normally you will have few different types of toxin, right? I'm just curious because when talk pathway, usually if we know that it's recognized some part of the peptidic and gluten from fungi up there by the secreted receptor, but how these toxin were recognized upstream there. Do you have any idea?
So this is one point I did not have the time to develop. We are working on it very actively. And the point is that we have the classical immune response where we're sensing directly these molecules from the cell wall, either peptidoglycan or beta-1,3 glucans, or even proteolytic activity from these fungi, for instance, some of these fungal proteases. But the point is that what we see, and we have shown it with restrictocin and also with vericulogen, just injecting the toxin alone is sufficient to induce an immune response that requires a toll pathway, but is actually non-canonical. So in the case of restrictocin, what we think is that we are triggering a response that is mediated either through the detection of the inhibition of translation, or actually there is this so-called ribosomal stress response. So we don't have really the data because we don't have the sensor yet for this ribosomal stress response, but it's clearly we find an involvement of the p38 MAP kinase pathway, and that would secondarily activate the toll pathway. So it's much more complex. So basically you have one layer of immunity where you're directly sensing the activity, the presence of structural components, and then you're triggering the classical toll pathway response. And then at a secondary level, you can sense the activity of virulence factors. And in this case, you're starting first through MAP kinases pathways and only secondarily through the toll pathway.
Thank you, Professor Ferrandon. Thank you for your foundational work using the traditional genetics in Drosophila. My question is about the future. Because do you, in your research, will incorporate the artificial intelligence of the machine learning to know about the complexity of the host and pathogen interaction?
Okay.
And the next question is from the evolutionary perspective. What's the pressure to drive the Drosophila evolve the multiple molecule of the vitamin A.
Yeah, of paramycin. Okay. So for artificial intelligence, I think you need to feed it data. And the problem is that we can describe the response in terms of Drosophila genes. But so far, we've not been technically able to really see which virulence factors are being actually secreted from the pathogen side. So it's something that is very difficult to do because we need to do it in vivo. And, like in the case of Aspergillus fumigatus, you have only 250 conidia per fly. So which means that you have a very, very low fungal signal. And we should probably try to do metabolomics or these kind of things. And we're working actually with the laboratory of Daniel Scharf in Zhejiang University to try to understand which toxins are important. Because what I showed you is that I've described restrictocin and verruculogen and actually fumitremor but Aspergillus fumigatus is secreting at least 26 families of secondary metabolites. And when we are making double mutants or triple mutants by removing also ergot alkaloids, we get still a very weak phenotype. So that means that we are actually missing most of the secreted virulence factors. And as long as we don't have this for the time being with artificial intelligence, I think it will still be a little bit limiting. But in the future, hoping that we get better at detecting the different things, if we can really go for metabolomics and doing also transcriptomics on the invading pathogens, and that would be probably very useful. And for the story about all of these effectors I was showing you in terms of evolutionary pressure, we find that these families are really restricted to Drosophila species. You don't find them anywhere else. But what I'm convinced of is that this thing, each organism must have evolved its response to adapt to this. And otherwise, I think all of these patients that are exposed to these mycotoxins, like I was saying from these former tuberculosis patients, they would die if there were no protection. But as I said, I'm not sure that there are adaptive immune responses against secondary metabolites. So I think most likely you have innate immune responses that are already been selected during evolution to protect against this kind of virulence factors. That's something that we don't really know. All I can tell you also is that, for instance, there is a family of antimicrobial peptides in vertebrates called beta defensins. So they are able to act on some microbes. But what has been shown some 10 years ago is that actually they are able to provide protections against pore-forming toxins. So again, it's a matter of having one antimicrobial effector that has a secondary function in protection against virulence factors just because of its biochemical structure. And it's fact that it's able actually to prevent the formation of these pores because of its amphipathic functions of the beta defensins. So, I mean, basically, there is a lot of adaptability of these molecules, and I think we are just really at the beginning of these studies.
So, may I ask the last question? So, the resilience is a very interesting idea, and I'm wondering whether you have some data to show that the activation of toll pathways can actually for the down-regulate the receptors to the mycotoxins.
Well,
because that may reflect the resilience. Because the toxin will somehow affect the cells, the host cells, and the toll pathway activation may down-regulate the receptors. That may be more like resilience.
Yeah.
Well, down-regulate some inhibitor factors.
Yeah. Well, I mean, inhibitor factors, you may have some. What we know is that we have, for instance, the activation of antimicrobial peptides is acting for only a limited period of time, and then after you have a decrease. But we don't really understand in terms of inhibition how it's working. So this may be the case, but I don't think we have really any hard data to answer it at this stage. But, and then, of course, for the toxins, we will need to identify the sensors. And we're starting to see it. But, C. elegans has really developed, this concept was developed of surveillance. And in C. elegans, what has been shown by Gary Rofrun, among others, who got a Nobel Prize a few years back, is that C. elegans is not able to sense directly infections. It's just sensing actually the activity of virulence factors that are targeting core biochemical processes such as translation, but also mitochondria. And that's something that is probably, and I would say highly likely to be also acting in our cells, because these are fundamental processes of life. And from the very beginning, when you think in evolutionary terms, when multicellular organisms developed, they were developing in a context of a huge excess of potential prokaryotes and potential pathogens. So it's something that has been developed from the very origin of animals, I think, and probably plants also.
Okay, thank you.
Science supports healthier, happier, and harmonious life.